All right. I know the long time from Sde Boker. We did our master's and PhD there. I mean, I did my master's there. Did you do your master's there, Udi? Yeah, you did.
Yeah.
No? Also master's, yes. We share the building, physics department. And yes, today I'm going to talk to you about early detection of soil-borne diseases with remote sensing technology. This research is supported by the head, how do you call it, Mad'an Rashi, principal investigator of the
Chief
agricultural
scientist.
ministry. Yes, and my partners are Mary Daphne Yelin and Ophir Dagani, two researchers in MOP Tzafon and MIGAL, who are phytopathologists, and On Rabinovich. And Moran and Gil are my research assistants that also did a lot of work. And let's start. So, why is it important? Soil-borne diseases like funguses that we're going to talk today are a significant threat to crop yield. In extreme cases, they can wipe out the whole field, and they're very dangerous, and they stay in the soil consecutively. So it's really hard to get rid of them. Once the soil is contaminated, then usually it's really hard to get rid of those diseases. Therefore, it is very important to give treatments, and treatments are usually with chemicals, pesticides, fungicides, and since most treatments are given as early prevention, therefore it's very important to detect those diseases early. Okay, if we are able to pinpoint to the areas where those funguses exist, then we can reduce the number of treatments. We're talking about very expensive materials, and we can target exactly the areas that needs the attention. And if we do it early enough, then we can prevent the spread of the disease, and that's what we want in order to increase the crop yield. So today we're going to talk about two diseases. The first one is called late wilt, or the pathogen name is Magna Proteopsis maydis. I hope I said it right. So this pathogen attacks corn, and it's called late wilt because it can be noticed only during the last stages of the crop. If we're talking about between 70 to 90 or 100 days of cropping of corn, so usually it appears only after 50 days. And then within 10 to 20 days, as you can see here on the left, it attacks the field and can really destroy the crop. It blocks um, the plant from transferring nutrients in the stem, and basically chokes the plant and kills it. And on the right, you can see that the corn cobs are gone. The other disease we're talking about is white mold, that is caused by the soil fungus Sclerotium wolfsi, and it's a multiserogate disease. So, whereas the late wilt only attacks corn, the white mold attacks many different crops, such as groundnuts, peanuts, tomatoes, carrots, and others. What it likes is heat and humidity. And here in Israel, during the summer, it's really hot, and in our region, it's also humid. And it's a very serious disease here in the Galil and also in other places in Israel. It spreads underground between neighboring plants where there is contact in the subterranean organs and grows. And again, the same trick. It blocks it attacks the root of the plant, and it blocks its nutrient transfer and symptoms are like dehydration and wilting. Here on the left, you can see the kishayon, the bodies of the fungus attacking the base, the roots. Okay, I'm going to talk to you a bit about the sensors we're using. Thermal imaging is one of them, a very important one. So every body emits long-wave radiation depending on its temperature. And if we're using sensors in the spectrum, in the thermal spectrum, between 7.5 to 14 micrometers, we can actually monitor the temperature of the object, in this case, plant. Okay, plants in water stress are warmer than non-stress plants. Why? Because when there is a decrease in water availability, the plant will protect itself and close its stomata. And then there is a decrease in the respiration rate, and the plant will evaporate less water. And because of that, there is an increase in the heat balance, because when a plant evaporates, it actually cools down. And this cooling doesn't happen, therefore, the plant gets hotter. So plants in stress are hotter. You can see here, there is can you see my cursor?
Yes.
All right, cool. So here you can see a blotch of a stressed plant. This is an RGB image. And here on the left, you can see it. Lighter color is hotter. Okay, and you can see the difference between soil and plant, also in temperatures. So that's one sensor. The other sensor we're using is a regular camera in the visible light. Okay, and so what does a camera enables us to see is the visible damage. Okay, but even more than that, we're using the GRVI plant index that relies on the differences in the reflectance of plant in the red and the green. Here it moved a bit, sorry, but this is the red right here, and the green is right where the red is. So actually, why are plants green usually? Because they reflect more of the green light. Therefore, what we see is the green color, their green color. Okay, and this is due to chlorophyll absorption bands. The chlorophyll absorbs more in the blue and in the red and less in the green. So actually, GRVI gives us an indication of higher chlorophyll content and more vigor. Sorry. So a plant in stress will have a lower GRVI than a plant that is healthy. Any questions so far? For me, I don't care if you want to ask during, as long as it doesn't interfere the flow. I welcome questions during my presentation. So the research goals of this project was firstly to develop a method for the early detection of these diseases, the white mold and the late wilt, both in corn and in peanuts and tomatoes. Okay, using imagery, remotely sensed imagery in the thermal and multispectral and visible range. And once we are able to detect it, we want to predict the rate of spread. Okay, that's the second goal. And the third goal was to examine the potential of identifying those areas, those hotspots, using satellite imagery in a large scale area. We are using mainly drones. Okay, so our methods were a periodic imaging of the area using drones or UAVs, unmanned aerial vehicles, with these sensors that I presented. And then we compared between different dates in order to see the change in the field. We had both commercial fields and experimental fields. So when you take images at the same place over time, you can see the difference, and then it is possible to detect the appearance and the rate of spread. Okay, And we also, of course, used in situ validation using a skilled field inspector that could tell us where there is a disease and where there is infection, okay? So we can validate our findings. And we did a comparison between our findings, namely the remotely sensed findings, and the in situ monitoring. And we also compared between our different sensors to see which sensor is better at early detection of these diseases. Okay, so this is one of our drones. This is the Matrix 600 Pro, but we also use DJI Phantom 4 and Mavic DJI Mavic 2, both with RGB cameras, and you can see their special resolution between 1 centimeter or half a centimeter to 4 centimeters in altitudes between 20 to 100 meters. And we use the Matrix 600 with several cameras. We use the Sony Alpha camera that is has a very high resolution. It could give us really close range, a very good spatial resolution. We also used ArgoWing multispectral lenses with 12 channels between 405 and 850 nanometers. And we used a thermal camera by FLIR, this altitude with a slightly less good spatial resolution, but still within less than 10 centimeters a pixel. We use Pix4D software both for autonomous piloting and also for georeferencing and mosaicing of the images. So today I'm going to talk to you about the two first seasons of the research. The project is a 3-year project, and we're still not done. We still have 1 more year to go. As I said, it was supported by the Ministry of Agriculture, and we're very thankful for that. And as you can see, the first season we did about 50 flight missions, and the second season we did around 130 flight missions, which is quite a lot for this kind of project. And we had 5 different locations, or 4 different locations. The Dash Farm, it's located in the Hula Valley, and we did we did a few experiments there with peanuts, corn, and tomatoes. I'm going to talk to you about it. And we also used our sensors in commercial fields. And for me, the real test is in commercial fields. If we are able to early detect the diseases in big scale as commercial fields, then we're actually making a difference. So I'm going to start talking about the methods now, and then we will move on to the results. One of the challenges is to mosaic images on top of each other accurately when we're talking about very high spatial resolution in centimeters. So if I want to compare between different campaigns, different dates, I have to put those layers on top of each other very accurate. And I have to do it because if there is any movement, then I'm actually comparing different plants, and this is useless. But we had some difficulties using our shelf software, the Pix4D mapper, because it created some distortions in this very high spatial resolution. And so what we did in order to have the maps accurately on top of each other, we had 2 solutions according to the scale. In small experimental plots like the Gadash farm, we disassembled the mosaic into small parts, 8 by 8 meters, for example, and then we accurately anchored the pictures on top of each other, and we used the ground control points for that purpose. Ground control point is a target that we put on the ground. We take its GPS with a handheld GPS that we have. It's an RTK GPS that has like a few centimeters, um, uh, very good resolution. And then we were able to put them on top of each other. But in big commercial fields, we cannot really do that. It will take ages and it's not feasible. So what we did that, we downgraded the spatial resolution, in this case to 25 by 25 centimeters. So these squares, we were able, as you can see here on the right, where is my, for some reason, here it is. So here you can see on the right, this is the original resolution, and it's within the altered resolution that we did. So that enabled us to put them one on top of the other. This picture is the GRVI image classification, and the bottom one, labeled C, is the thermal sensor. This also gave us some advantages. Here you can see that this pixel, for example, has mainly soil and some vegetation. So we were able to use thresholding to discover those mixed pixels and to omit them from the calculation. And we only took into consideration pixels that had vegetation. Okay, in order to make sure that our thermal sensor works properly, we used a portable meteorological station that we took with us to the field that had temperature, wind direction and intensity, and radiation intensity measured during the flight. And we also had two reference bodies with a wet and a dry reference bodies that we measured their temperature, and then we compared them to our sensor. And before and after going to the field, not every time, but sometimes we use the black body in order to make sure that our thermal camera is calibrated. Okay, so the algorithm that classified stress was dependent on the air temperature in the field at the time of the imaging. So for the thermal classification algorithm, we classified the pixels into 6 different groups. Okay, starting from very healthy to healthy, less healthy, stressed, severely stressed, or dead plant, and soil. Okay, pixels that had a temperature over 10 degrees Celsius over the air temperature were classified as soil, and we took them out of the calculations because we're not interested in looking at soil, only at the vegetation. The 15 pixels with the lowest temperature were classified as a very healthy plant, and then the rest of the pixels between these two edges were divided into four equal groups with equal temperature intervals. Okay. So when we come to validate land cover, we cannot really do field monitoring at the plant resolution, okay? And in large scale, in plot scale, it's really impossible. So what we did is that we used RGB image as the base image for classification validation with the aid of two additional plant indices that we use that are based on the RGB channels. These are the GLI index and the VARI index. Both of these indices, different than the GRVI index that I presented, also use the blue channel. You can find their formula in many articles. But these two additional plant indices helped us to distinguish between soil and plant. We use the GLI index to filter out the soil, and we use the VARI index to classify between a living plant and a dead plant. The GRVI had difficulties in distinguishing between a dead plant and soil. So here, as you can see, on the top, this is the area of the peanut plot in the Dash farm from 2019, where we did the classification validation. The purple rectangle is around 15% of the area, and here it is enlarged. This is the RGB image. And here in image C, you can see the difference between a dead plant and a living plant. Soil was filtered out. All the white spots are soil, and the red areas are dead or very stressed plants. But as you can see, it's not 100% because the mixed pixels that are on the edges are also red. And it's actually not a dead plant or a stressed plant. This is an artifact of a mixed pixel. So we also use some filters to distinguish these thin streaks and omit them. Here you can see different indices that we use to classify. So D is the vari index. E, if I'm not mistaken, is the right. What does it say? Baseline. C is the baseline image that we used for the classification, for the comparison. D is the GLI, and E is the vari. F is the GRVI. That's the one we use most. And G is the thermal sensor. Okay, and you can see that all sensors and indices are doing pretty well in classifying stress. Some of them are overly classifying stress, meaning different degrees of stress. You can see at the RGB that, yes, there are more stress. The differences in colors can distinguish, can show us different levels of stress. Here in the thermal sensor, however, it's purely based on temperature. And as you can see, the thermal sensor is really good in really showing the stress plants when you compare it to the base image. Okay, we use the confusion matrix to measure or validate our findings. I'm not going to go over this. I'm pretty sure most of you know what the confusion matrix is, but just to reiterate a few terms, overall accuracy is actually measures the ratio between the correct identification of our sensors and the total population of objects. So if we're talking about this is the remotely sensed classification and this is the manual classification. So this is predicted positive or negative, meaning a healthy plant or a stressed plant. So total overall accuracy, for example, is what we predicted positive and it is positive, and what we predicted negative and it is negative, divided by the total population, meaning taking into consideration false positive and false negative as well. User's accuracy is called precision, and it predicts reliability. It's actually telling us whether an object that we marked on the map, on our map, the sensor map, will actually be present in the field. For example, if I say, here there is a stress plant, go to the field and find it, what are the chances that if you go to the field, you actually find it where I told you? Producer's accuracy is the probability that an object that I see in the field, like an inspector sees in the field a stressed plant, what is the probability that it will be correctly classified on my map? And kappa coefficient evaluating how well the classification performs when comparing to just random assigning values. So our classification had an overall accuracy of over 94%, with users and producers accuracies for plants really high. For soil, also pretty high. But for dead plants, we had some difficulties, as you can see here. Another thing that, that we wanted to check, sorry, in our goals was to see if satellites can really distinguish and can really pinpoint to areas with diseases. So here you can see the commercial field of peanuts in different dates. And the top image is the RGB image of our drone. The middle panel is when we did the GRVI classification, and the lighter dots is the area with infection. And you can see it pretty well with the UAV using the plant stress index. And as you can see, the infection rate increases along the dates. But when you look at the satellite imaging, and this is Venus images with pixel size of around 5 meters, you cannot really see any change. And if you look at the numbers, the Venus, you see that the mean NDVI of Venus stays high throughout the whole season. There is no change. And there is actually no standard deviation, meaning most of the pixels are healthy according to Venus, which showed us that satellites at this special resolution, the free satellites that we're using today, like Sentinel, Venus, and of course others that are free, are not good enough for this project. So that was sad, but then in 2020, we did not use satellites anymore because we realized it's only when there is a very big, massive spread of disease, that's only the possibility to see it with satellites. But by that time, the farmer has lost most of his crop, and the farmer will be able to see it by himself. He won't need the satellite. Okay, let's move to some results. These graphs show the comparison between in situ infection inspection and our sensors stress and vegetation cover ratios. So here you can see in different dates, this is the field in a commercial field of industrial tomatoes. You see that the orange line is the infection rate measured by an inspector, and the black line is the stress rate measured by our RGB sensor, and the gray is the cover, the plant cover in the field, also measured by our sensors. So as you can see, that the plant cover, as expected, rises until it reaches an area when the infection rate goes up, and then the cover drops. And also you can see an increase in the stress rate from our sensors. So here there is a bit disagreement where we show that there is a decrease in stress and an increase in cover, whereas the inspector sees a constant, relatively high infection rate. But here there is an agreement. Oh, sorry. When we move to these graphs on the bottom, we see a scatter plot between the infection rate measured by the inspector and our stress rate based on the RGB sensor. And you can see that there is a correlation, not very strong, but 0.74, R square value of 0.74 is pretty good for a field study in a commercial field. You can see that the agreement decreases over time. And this graph shows the comparison between our thermal sensor and the infection rate. And it's also relatively, it's okay, I mean, not super great. Another project we did in Yavne in a commercial field, but this is an experiment. It's a cultivar experiment. Did I skip? No. We checked 10 different or 12 different cultivars of corn. Some of them were susceptible to late blight, and some of them were more resistant. And we took images, both in RGB and thermal images, in 2 dates, 73 days from sowing and 94 days from sowing. And here, this is a close-up on block number 4. You can see that here, not so much stress, but here in day 94, you can see areas that are stressed, and you can see it in both sensors, okay? This is actually now in review, and hopefully we will be able to publish these data. So we did a validation of our findings versus in situ findings. So what we did, we took these 2 blocks and we marked the areas that were sick. And then we ran our sensors, both GRVI and thermal ones, and we got overall accuracy of over 90%. Cup coefficient is a bit lower, and this is probably due to high false positive rates, meaning we say there is stress in areas that were not sick. But most of the areas that were sick, or almost all of them, were identified by our sensors. We did it in a different way, where we took the whole field and we randomly selected 1 by 1 meter units, covering 20% of the cropped area. And we did the same exercise, meaning we manually detected the disease in these units, and then used both the RGB and the thermal sensors to validate the results, the in situ results, and we got similar overall accuracies over 90%, which is pretty good. Here you can see that overall accuracy is around 93%, and we are doing really good in identifying healthy plants. What we are not doing so great is identifying stress plants, okay? Because some of the stress plants that we say are stressed are actually healthy, and we have a high false positive rate that actually damages our findings. But this is an issue that is debatable. Here I'm going to show you another hit-miss ratio that we did in a commercial field with peanuts here in Hula Valley. So what we did here is we did a comparison between our sensors and in situ inspector that went in the commercial field. We chose 8 different rows, and over 30 meters, the inspector went every meter and gave his marking for inspection if that area is healthy or sick. Now, we did this in 10 different dates. We took an image at a certain date, and the inspector went on that date and checked all these 8 rows. Now, we both had our grades, our markings. In areas where our sensors discovered stress before the inspector did, let's say we discovered it in date X, and the inspector in date X didn't see the plant didn't seem to be stressed or sick or infected, but in date X+1 or X+2, it was indeed infected, then it was actually, we were able to see the disease before it was seen by the naked eye. So we marked it as a hit with the yellow color. The opposite was when the inspector saw the disease and we didn't see it, then it was a myth. In areas where we agreed, what was an agree? That at the same date we said that it is healthy, and it was indeed healthy, and stayed healthy throughout the whole season, then it was agreed healthy. If both of us, meaning the sensors and the inspector, discovered the sickness at the same date, then it was agreed sick. Okay? And we did it in two different sensitivities. One of them was 2.5 degrees Celsius gap between the, remember we talked about the classification with 6 groups, and we did the gap between the groups if it was 2.5 degrees or 3 degrees. What is the difference? When the gap is 2.5 degrees, then the sensor is more sensitive, meaning in lower temperature, it will already show stress. In 3 Celsius degrees difference, the sensor is less sensitive. Now here you can see the differences between ground monitoring and RGB sensor, and between ground monitoring and the thermal sensor. So the heat rates were higher, as you can see in the 2.5 degrees Celsius gap. 1 second. Sorry about that. So in 2.5 degrees Celsius, our heat rates were higher than in 3, but also the false positive rates were higher. When we did the 3 degrees difference, we did not have any false positive, but our heat rates were lower, both in RGB and in the thermal sensor. Our miss rates were relatively low in both, but lower in the higher sensitivity. So this is some kind of a trade-off. Usually what farmers, what they prefer is not to miss areas that are sick. So they don't care if there is a low or false positive. I mean, as long as I don't miss areas that are sick, they will be more satisfied, because then they can go and spray the whole area. And if they sprayed some of the area that was not sick, it's still better than spraying the whole field. But then again, you don't want to be giving false positives in a higher rate. So this is something to consider. Okay. Another similar check that we did was in 2020. This is the second year of our research. Early detecting of wild mold in a peanut commercial field. Here we did the same. We had 8 rows that we divided to 1 meter by 1 meter plots, and each row was 20 meters length. So we had 20 times 2 times 8, around 320 or 480 plots. And we did the same. We took images in different dates, and at similar dates, we had an inspector going and marking and saying how many plants are sick in that 1 meter by 1 meter plot. This is the thermal sensor. As you can see, this is a highly infected area. And this is the classification of the thermal sensor. The heavily stressed plants are marked in red. And this is the in situ field monitoring. So in 1 by 1 meter plot, there are usually around 4 plants. And if we had 0 plants that were sick, then it was 0 infection, 1 plant 25, and so on and so forth to 50, 75, and 100%. And then we did a comparison, okay, between our findings and the in situ findings. So as you can see here, out of 320 plots, 209 plots, we discovered the disease earlier than the inspector. 56 plots that were false positive. Only 4 plots we missed. And as you can see, agreement was in 51 plots. There was no agreement about healthy because most of the plots were actually sick. So as you can see, this is a very high hit rate. Not going to go into the details of how we did the thresholding, but whoever is interested, I can talk about it later. One thing I want to show you about using our multispectral camera. This is the same peanut commercial field. And here you can see an area that was infected. So when you look at NDVI, you can see that NDVI is not really sensitive to soil. I mean, it looks at soil and also at areas that are sick and gives them the same. Here, this is a sick plant, but it gives it the same grade as soil. GRVI is slightly better. It gives slightly better, but if we look at our multispectral cameras and the ratio between 650 and 405, 405 is in the blue and 650 is in the red. This is actually deep blue 405. And you can see that it really gives good distinction between dead plants here, and it can really filter out the soil. So this is something that we want to look more in depth at the 405-650 ratio. Here we are trying to, this is also one of our goals, was to actually analyze the rate of spread. And this is something I want to show you. This is not quantitatively, but qualitatively. You can see rate of progress of disease in an experimental peanut plot. You can see the dates. Now here you can see in August starting an area that is starting to be infected and spreads out. Okay. Now the same thing you can see with the thermal sensor, but the thermal sensor sees it in advance. Here, September 4th, the area that is sick is small, but the thermal sensor sees already a higher rates of infection here as well. What we see on September 4th with the thermal sensor, we see with the RGB sensor only a week later. And here, what we see on September 11th, we only see in October 6th with the RGB sensor, meaning that the thermal sensor is able to see the stress and the sickness before we can actually see it with the visible naked eye. That same peanut field that was highly infected in 2020, we, as I said, took images in 8 or 11 different dates. Here I'm showing only 8 different dates. And the infected areas or the stressed areas with the RGB sensor are the orange and red colors. As you can see, this is an area that spreads out. There's actually 4 different areas that had disease. And what we did here is we marked the epicenters of the stress according to dates. So every color represents a date that was an area that detects an area that was infected, meaning the first time that this area was infected was marked with that color of that date. And as you can see, this is an area, the light green is where the first time we took, and then light blue and slightly, and as it grows in space, we were able to measure an average spread. So in the direction of north-south, we saw a spread of half a meter, more than a bit half, 0.55 meters per day averagely. And in the east-west, we saw a similar but less, bit less spread. Now, what's interesting is that this area was irrigated by a linear move irrigator, and the track was moving from north to west and north to south. And this direction, the disease spread faster. So maybe the irrigator has something to do with the direction and the speed of the spread. This is something we want to continue investigating in our next final year of research. So to summarize and what we talked about, our proposed classification technique of looking at the 15% of coldest plants and looking at the air temperature plus 10 degrees Celsius has proved successful in classifying the state of stress and plant health, as was then validated with in situ monitoring. And the thermal sensor could detect the disease even before it was seen by the naked eye or by the RGB sensor or the inspector. If you remember pictures I showed you, and the high heat rates of both sensors. So there is a trade-off between the ability to detect early diseases with a low miss level that come at the expense of a high level of false alarms. That trade-off I showed you, and this is something to consider, but we have the ability to fine-tune our models and according to the needs. Okay, right, like I said, we are able to refine those findings. It's difficult to distinguish between a dead plant, shade and shadow with the GRVI classification, but we got improved classification using the GLI and VARI indices, and we plan to use them in the future, this combination of vegetation indices. I didn't talk about it, but we used the blue channel to eliminate bare soil. What happens is, in most cases, when we take an image of a field, we get two peaks in the blue channel. The first peak is plants or vegetation cover, and the second peak is soil. Now, if we use that blue channel and we omit that second peak, we're left only with the plants. And this is another reason why GLI and VARI are better in distinguishing between soil and plants than the GRVI, because they use the blue channel and the GRVI doesn't. So channel 405 is promising in terms of detecting dead plants. We're planning to look into it more in the future. Satellite imager did not prove successful in locating outbreaks of soil diseases. Our solution of downgrading the spatial resolution, converting the resolution into larger pixels proved to be successful in our improved ability to anchor images on top of each other. This is usable when you need to compare between different dates and to be able to pinpoint that area and not having mistakes. Monitoring plants over time makes it possible to identify hotspots of disease and to determine the rate and the direction of spread of the disease. So it was hard for us to detect the rate of spread because there's many parameters that are involved that influence the spread of the disease. We're still working on it. Hopefully we'll be able to show better findings in the future. So future work, we want to integrate machine learning. We've been using machine learning technology and algorithms in other projects, and it proved really good. I'm not going to talk about it today, but we hope to use that same technology to classify, to show the computer how does a dead plant or a sick plant looks like, and to train the machine to classify it by itself. We are planning to get better at forecasting rate of spread. And we recently purchased a hyperspectral camera, and we want to use it to better identify the disease. I want to thank you. I hope it was interesting for you, and I want to thank all the large team of researchers and farmers and growers and inspectors, in situ professionals that helped us in this project. And this is my email address, and you can be in touch with me. I would love to cooperate in many fields. And I would like to thank you for your listening. And if you have any questions, I hope we have time.
Yes. Thanks, Assaf. Thanks for the nice presentation. Any questions? We have several minutes for questions. You can just unmute yourself and ask.
I have a question, please. May I ask a question?
Yes, sure.
Yes.
In the end, you said about significance of the channel of 405 nanometers, right?
Yes.
And also in the very beginning, as far as I understood, I was discussing with the index, red-green index.
Yes.
And then also the red wavelength, something like about that. And you said that green is related to chlorophyll.
Right.
But what is responsible for the processes in the red area, and why it's 405 nanometers? And
okay,
what kind of the chemistry is behind of that? If it's
I'll try to make a bit order in this. So the red is in the 600, 620, and chlorophyll absorption band. There's chlorophyll A and chlorophyll B. Their absorption band is in the red and in the blue.
Okay.
The green is less absorbed by chlorophyll. So the difference in absorption between red and green allows us to show chlorophyll rates.
Okay.
So higher chlorophyll, meaning there is more absorption in the red and less absorption in the green, the GRVI will be higher, because GRVI is actually red minus green divided by red plus green. So the difference between red and green, the higher the difference between red and green, meaning the more chlorophyll there is in the plant. Now the blue is also absorbed by chlorophyll, and I think it has to do why blue is also beneficial in this kind of way. The blue helped us to distinguish between plant and soil, not because of chlorophyll, but because soil tends to have higher, how would I say, it reflects the blue more, okay? So albedo, soil albedo, and properties of soil make it that the reflectance of the blue is higher in soil than in plants. And we use that to eliminate soil. I hope I answered your question.
Okay, thank you.
Could I ask a question?
Sure, yes.
Okay. So you talked about, I have a few questions. You talked here about 2 field crops. Have you attempted and explored looking at tree crops?
We have projects with orchards, but it's not with these soil-borne diseases. It's with other diseases like, how do you call them? Asha Charuv in almonds, and Tzirat Shaked, also in almonds. And over there, we are not looking at the effects of the disease, but we're trying to look at the almond fruits. But to your question, we do have, we just got funded to look at the akariot. How do you say? How do you say?
Right. Mites.
Mites, yes, spider mites
Yes.
in apple orchards. And here we're going to use hyperspectral imagery, which is slightly different. It's way more sensitive. So yes, we do look at the orchards as well, but it's in a different way.
Okay, and I also had another question, which, so in your introduction, you said that, you know, one of the ways that you use the temperature to evaluate this water stress has to do with, because the plant will close the stomata. And I was wondering then, in plants that actually don't control stomata at all, I suppose that this is simply not useful. That's a question. And then my second question also has to do with the question of chlorophyll and green color on their plants whose actually their flushes, the new leaf flushes are red rather than green, and they only green upon maturing, but they're very healthy. They're actually flushing. So I was wondering if you've struggled with some of these aspects.
Okay, so all right, your first question was about stomatas, and yes, indeed, let's for example, citrus trees do not really close their stomatas, so it's really hard to see stress in citrus with thermal sensors. So yes,
Okay.
it doesn't work for all plants.
Okay.
And with what you said about the leaves, so yes, leaves have pigments, and sometimes the red pigment, the anthocyanin, is more expressed than the chlorophyll. But mostly when the chlorophyll is expressed, it hides the effect of the other pigments. Like when leaves get older, they have less chlorophyll. That's why they turn red, because now the chlorophyll is gone and the anthocyanin is now more expressed. But also, I don't know, I haven't had a research with plants that the leaves start red, but I guess Neshirim, some of them, like Shazif, like plums, start red and then they turn green, right?
So does cocoa.
Okay, I assume he will have difficulty with GRVI and this kind of chlorophyll-based vegetation indices, and in these cases we will have to use other indices, and we have to take into consideration phenology and our knowledge. That's why it's very good to surround yourself with people who know about plants. And in my case, I have people who know about plants and can tell me, listen, here you need to do something else. Since I'm not a plant person, it's really important to have somebody that really is an expert and knows to tell you so you don't make silly mistakes.
Okay, thanks.
Thank you.